dietary fiber, which is classified as soluble and insoluble fiber. In this particular module, dietary fiber, we will be learning about cellulose, hemicellulose, lignins, gums, mucilages and pectins. Impact of dietary fiber on health is also determined by its physiochemical properties. Hence, we will also be learning about physiochemical properties of fibers like porosity, viscosity, solubility, to name few. We will also be learning about different sources of fiber and the absorption and binding of different ions. Let us now see each of the dietary fibers in brief. First, cellulose. It is a major cell wall component in the plants, an unbranched linear chain of several thousand glucose units right from
beta-1,4 glycosidic linkages.
Cellulose mechanical strength. It is resistant to biological degradation. It is low aqueous solubility and resistance to acid hydrolysis result from hydrogen bonding within
microfibrils.
Aspinall in 1970 studied that cellulose is insoluble in strong alkali and there is a portion of 10 to 15% of cellulose referred to as amorphous, that is more readily acid hydrolyzed. Cellulose are not digested to any extent by the enzymes of human gastrointestinal system. Second is hemicellulose. These are cell wall polysaccharides solubilized by aqueous alkali after the removal of water soluble and pectic polysaccharides. They contain backbone of glucose units with beta-1,4 glycosidic linkages but differ from cellulose in that they are smaller in size, contain variety of sugar and usually branched. They contain mostly xylose and semi-galactose, mannose, arabinose and other sugars. Lignins. It is not a polysaccharide but a complex random polymer containing about 40 oxygenated
phenylpropane,
including coniferyl and alcohols that are undergone a complex dehydrogenative polymerization. Lignins vary in their molecular weight and
methoxyl
content due to strong intramolecular bonding which includes carbon-to-carbon linkages. Lignin is very inert. Lignins demonstrate a greater resistance than any other naturally occurring polymer. Let us now understand pectin.
Pectic substances
are a complex group of polysaccharides in which
D-galactronic acid
is principal constituent. They are structural components of cell wall and also act as intercellular cementing substance. Pectin is highly water soluble and is almost completely metabolized by colonic bacteria. Due to their gelling behavior, the soluble polysaccharide may decrease the rate of gastric emptying and influence small intestine transit time. This explains their hypoglycemic properties. Let us now learn about gums and mucilages. These are the type of plant fibers that are not cell wall components but are formed in specialized secretory plant cells. They are reported to be highly branched polysaccharides that are formed gel, bind water and other organic material. Guar gum is isolated from the seeds of guar. Partially
enzymatic
hydrolysis result in the product that can be used as a soluble dietary fiber. The physiological effect of this fiber source comply with the water which might be expected from the soluble fiber. Gum arabic is excluded from Acacia trees. Since we have seen the classification and the properties of all the soluble and insoluble fiber, let us now understand the physiochemical properties of dietary fiber in general. Dietary fiber is a complex mixture of polysaccharides with many different functions and activity. It passes through the gastrointestinal tract. Many of these functions and activities depends on their physiochemical properties, which are now described. First is particle size and bulk volume. The particle size plays an important role in controlling number of events occurring in digestive tract, that is transit time, fermentation and fecal excretion. The range of particle size depends on type of cell wall present in food and their degree of processing. Particle size of fibers may vary during transit in digestive tract as a result of chewing, grinding and bacterial degradation in large intestine. Beyond 550 millimicrons, the hydration properties were found to decrease with decrease in particle size during grinding. The fat absorption capacity was also reported to increase with decrease in particle size. Let us now understand the surface area characteristics. Porosity and available surface can influence the fermentation of dietary fiber, while regiochemistry of the surface layer may play a role in some physiochemical properties. The porosity and the surface available for the bacteria or molecular probes such as enzyme will depend on architecture of the fibers, which is related to its origin and processing history. The third property is hydration properties. The hydration properties partially determines the fate of dietary fiber in digestive tract and account for some of their physiological effect. Swelling and water retention capacity provide a general view of fiber hydration and will provide information useful for fiber supplemented food. Process such as grinding, drying, heating and extrusion, cooking are the examples which modifies the physical property of the fiber matrix and also affect the hydration properties. The environmental conditions such as temperature, pH, ionic strength, dielectric, constant of the surrounding solution and the nature of ions can influence the hydration characteristics of fiber containing polyelectrolytes. Let us now understand the solubility and viscosity. Solubility has a profound effect on fiber functionality. It is also well established that soluble viscous polysaccharides can impede the digestion and absorption of nutrients from the gut. If polysaccharide structure is such that molecules fit together in crystalline array, the polymer is likely to be energetically more stable in the solid state than in the solution. More branching, presence of ionic group and the potential inter-unit positional bonding with mixed increase the solubility. The viscosity of the fluid can roughly described as it is a resistance to the flow. Generally, as a molecular weight, the chain length of the fiber increases. The viscosity of the fiber in the solution increases. However, the concentration of the fiber in the solution is also affected. Absorption and binding of ions and organic molecules also affects the fiber properties. Fiber has been suspected of impairing mineral absorption because of charged polysaccharides such as pectins through their carboxyl group and associate substance such as phytates in the cereal fibers, which has been shown in vitro to bind the metal ions. Charged polysaccharides do not have effect on mineral and trace element absorption, while associated substance such as phytates can have negative effect. The environmental conditions such as physical and chemical form of fiber and the nature of the bile acid may influence the absorption capacity of the fiber. Let us now see The sources of dietary fiber. The dietary fiber is naturally present in cereals, vegetable, fruits and nuts. The amount and composition of fiber
range
from food to food. It is suggested that healthy adult should consume between 20 to 35 grams of dietary fiber each day. Different non-starchy food provide up to 20 to 35 grams of fiber per 100 gram dry weight, and other containing starch provides approximately 10 grams per 100 grams of dry weight, and content of fiber of fruits and vegetable differs from 1.5 to 2.5 gram per 100 gram of dry weight. The fiber content of commonly found Indian foods are presented in the table, which is mostly seen. The common highest containing fibers are barley, corn, oats, rice, then even wheat germ, legumes, green beans, peas, kidney beans, different types of lentils, bitter gourd, fenugreek, lady's finger, tomatoes, eggplant, cucumber, cauliflower, different types of fruits like unpeeled apple, pineapple, pomegranate, strawberries, peach, and nuts like almond contains high amount of fiber. Coconut raw does contain good amount of fiber. Peanut contain good amount of fiber, and the flax seeds, which contains approximately 22.3 grams of fiber per 100 grams. Let us now understand the effect of processing on dietary fiber content of food. The physiochemical properties of fiber can be influenced by diverse treatments like chemical, enzymatic, mechanical, thermal. Thermomechanical to process their functionality. For the example, mechanical energy have a profound effect on polysaccharides, as reported by Ffn et al. In 1998. Grinding may affect the hydration properties, in particular the kinetics of water acceptance. As a result of increase of surface area, fiber hydrate more rapidly. Heating generally changes the ratio of soluble to insoluble fiber. Combination of thermal mechanical energy can change dramatically the structure of dietary fiber at all structural level, leading possibly to a new functional property. Processing required to make some vegetables, legumes suitable for eating, because it causes a decrease of several components of fiber. For the example, during cooking of lentils, previously dipped, the quantity of fiber diminished fundamentally to a greater decrease in hemicellulose, as suggested by Tatzana et al. In 2002, and further their study stated that modification that happens during the thermal processing, the kidney beans that are reported solubilization of polysaccharides resulted in decrease of total fiber content, and mainly due to loss of soluble fiber.
Penner and Kim in 1991 analyzed the non-starch polysaccharide fraction of raw, processed and cooked carrots. Result shows that processing and home cooking of raw carrots showed an increased amount of non-starch polysaccharide units per dry weight.
And cooking of canned carrots resulted in large increase in total and soluble non-starch polysaccharide unit. Dry weight were not as great when compared on weight basis. Chopper et al. In 2009 studied the effect of soaking on soluble and insoluble and total dietary fiber of Bengal gram, cowpea, dry pea, field beans and green beans. Samples were soaked in tap water for 12 hours at room temperature. Soaking increased total dietary fiber by 1.2 to 8.2%, and considerable increase in soluble dietary fiber was observed. Here we come to an end of the effect of processing, structure and of course the sources of dietary fiber. Let's conclude. Learners, hope by now you must have understood the important role of properties of fiber in imparting health benefits. This approach recognizes the diverse and variant action of each dietary fiber, which may be modified as a result of processing and cooking. This particular module explored more systematic way how physical properties affect the fiber performance in imparting health benefits.